Um, thank you again for being part of this discussion tonight and this conversation. We're happy to provide this COVID-19 resource to all of you. As a reminder, this event is for informational purposes only. For specific questions and concerns about your health, please speak with your personal physician. And again, just an FYI that we are recording this event. Once we get started, please make sure you are muted, but feel free to unmute yourself and ask a question or a follow-up. And remember that you can always use the chat for questions as well. So we're going to keep this to about 45 to 50 minutes tonight. So we're going to jump right in. And as people come on, we'll just let them hop right in. So I'm going to turn it over to Lucas.
Well, I want to echo Trish's welcome, and I'm glad to have you all with us. I expect our group will grow a little bit as we begin, but I want to start out by introducing to you Tyler Martin, my friend and a great alum of UNK. He's doing some very, very interesting work, and I think we're excited for him to share with you. Dr. Martin's a 1981 graduate of Kearney State College. He's the CEO of Adjuvants Technologies based in Lincoln. He's a Hebrew native, holds a degree in chemistry with a biology minor. He earned All-American honors in track as a loper, and his name can still be found in the Foster Field Outdoor Record books. I know he's proud of that, and he should be. It's been a few years. Some of you may remember him as Tyler the Half Miler, actually, because of his running acumen. He's a UNK Alumni Association Distinguished Alumni Award recipient, as well as Dr. Don Fox lecturer, and he's spoken several times on campus. I'm interested, maybe raise your hand if you were in school with Tyler. There's probably a few of you on there who came just to see, does he look as good as he did when he was in school? I'm sure the answer is yes.
Got a thumbs up out there, I see that.
Dr. Martin's a 1986 grad of University of Nebraska Medical Center. He was a resident in pediatrics at UNMC and was recognized as the outstanding student in pediatrics. He was a postdoctoral fellow in molecular microbiology and pediatric infectious diseases at Washington University in St. Louis. And he was named outstanding teaching fellow, St. Louis Children's Hospital in 1990. He also served as an NIH-funded vaccine investigator, including COVID vaccines from 2015 to 2020. So with that, I'm going to turn it over to Dr. Tyler Martin. Take it away, sir.
Thanks, Lucas. And thanks for writing that awesome story about me. I don't know that I could have told a better story myself, so thank you. Guys, it's really nice to be with you all tonight. I see some names that I know on the chat box, and wish we could be together, but of course that's not going to happen this year. So I thought I'd just start off by helping you all understand one other point about why I know anything about this particular topic. As Lucas mentioned, this company has received funding from NIH to look at COVID vaccines. One of the approaches that we're taking back to these COVID vaccines is to use vaccine adjuvants. And there have been 4 adjuvants that have ever been approved by the FDA. Teams that I led when I lived in California developed 2 of those 4. So this is kind of my area of expertise. So I thought to help orient you all to this situation that we find ourselves in now about trying to make decisions about which vaccine should I take? Should I take it? Should I not take it? And just be helpful to kind of review how we got to where we are. So COVID-19 is our name for the disease that's caused by the virus that we call SARS-CoV-2. So you'll sometimes hear those words used interchangeably, but they're actually not. When we say SARS-CoV-2, we mean the virus. And when we say COVID or COVID-19, we mean the disease that's caused by that virus. The disease was first recognized in China in November of 2019. We don't have any knowledge of where it might first have occurred, but it was first recognized in China in November. The sequence was published in January of 2020, so essentially one year ago. And when that sequence was published, and so by the sequence, we mean the genetic sequence of the virus. When that sequence was published, that began the worldwide race to develop global vaccines for SARS-CoV-2. So I'm going to pause that story there for a second and now move to a story of how do we normally develop vaccines. You may have read in the newspaper that it, or heard on the news that it often takes 10 to 20 years to develop a vaccine. But yet here we are 1 year later with vaccines that are available. So I want to sort of explain how that normally works. When we normally, or in a routine situation, when we develop a vaccine, we're not really facing a global pandemic. Somebody gets an idea that, hey, I can prevent this disease that happens, to a few people every year. And so I'm going to start working on that. And I first do a phase 1 study. That usually takes about a year. I look at the results. I figure out how I want to do my phase 2 study. That's going to take me 2 years. I take another year to look at the results. Then I do a phase 3 study, which is what the ultimate study that the FDA needs for demonstration of proof. That usually takes about 3 years. Then it takes a year to write up the results, and it takes a year for the FDA to review it. So that's kind of the best case scenario, is sort of a 10-year process. And at the end of those 10 years, the vaccine has typically been used in between 3,000 or 10,000 patients. It kind of depends on the target that one is looking at, but 3,000 to 10,000. Those patients have all been followed for usually about a year after they got their last dose of vaccine. So we have about 12 months of safety follow-up from the time the last patient was treated. And all 3,000 to 10,000 of those people will have what we call tolerability data. So that is what happened in the first 7 days after they got vaccine. Did their arm hurt? Did they feel sick? Did they have a headache? Things like that. So just remember those points, because we're going to come back to that later. So 3,000 to 10,000 patients, everybody has 7-day tolerability data, and everybody has 12 months of long-term safety data at the time that vaccine gets approved. And when vaccines are approved, they're approved based on 3 or 4 different attributes. So if you have a disease that you can measure prevention of that disease, we measure prevention, and that prevention is called efficacy. So you can get an approval based on efficacy against a certain disease. Some diseases don't happen very frequently, and so you get your approval based on generating an immune response to the vaccine target. And we call that approval an approval based on immunogenicity, which means the ability to induce an immune response. All vaccines get assessed for tolerability, and then all vaccines get assessed for safety. So that's the normal story that we routinely use. And now I'm going to go back to January of 2020. When this whole thing happened, it created this race for 4 different types of vaccines that get put into development. The first one that I'll mention that we don't have yet today are the traditional vaccines. Traditional vaccines, you take the DNA sequence of the virus, you move that DNA sequence into a producer cell. That producer cell then makes the protein that you've taken from the virus that the DNA codes for. So the DNA makes RNA, the RNA makes protein. We take the protein out, we combine that protein with an adjuvant, and we treat people. The longest part of that process is making the cell that's going to make the protein. That typically takes about a year. And that's why the companies that are making the traditional vaccines are so far behind the other companies, because it takes a year to make that producer cell. All the rest of the steps are the same. Phase 1, phase 2, phase 3, they're all the same. We're just doing them sort of overlapping rather than in sequence. And we'll come back to that story again later. The second group of vaccines, which are the ones we have today, are these RNA vaccines. There are 2 that are currently approved in the United States. One is manufactured by Pfizer in collaboration with a German company called BioNTech. And the second one is made by a company called Moderna. Now, when this whole thing first started, some of you who are friends of mine may remember that I was putting out these kind of weekly updates on what was happening with vaccines. And I said when this started, I did not think that the RNA vaccines would probably work. They're very, very fast, but I didn't think they would work because they have never worked before for anything that we've ever tried to use them for. Fortunately, they have worked. And they are so fast because all you need to do is make this genetic sequence. You don't need to move that sequence into a cell. The way we use them is, remember I talked about DNA goes to RNA, goes to protein. So in this case, we short circuit that entire process and give you the RNA. And so you make your own protein. So instead of me having to make a cell that makes the protein and purify that protein and give it to you, we give you the RNA instructions so that your cell makes the protein. And it's why it's so very, very fast and so much faster than anything else we've ever done. In fact, it was so fast that those companies were in the clinic treating human beings in March of this year. So sequence identified in January, treating people in March. That's never happened before. And it's because this technology is so fast, because you don't have to make the protein, you just have to make the instructions for the protein, and then our bodies manufacture the protein. The next group of vaccines, which actually started at the same time and moved as quickly, but they haven't been as effective, are DNA vaccines. And the leading company in the US that makes the DNA vaccine is a company called Inovio. Inovio was, again, as fast as the RNA vaccines because DNA is very, very much like RNA. But Inovio has also never had an approved product, and their product so far has not shown the right immunogenicity to make us think it might work, and so they're just not making very much progress. But they were very fast, and at the beginning, we're right there with the RNA vaccines. And then the fourth type of vaccines are adenoviral vector vaccines. Adenovirus is a virus that we commonly get it's one of the cold viruses. When you get a cold that also includes conjunctivitis or pink eye, that's usually an adenovirus, whereas regular colds are usually caused by different kinds of viruses. That's not a completely true statement, but it's generally the case. If you have pink eye, you probably have adeno. And there are two types of adenoviruses that have been developed. One is an adenovirus that a company called Johnson & Johnson is developing. That is a human adenovirus. It's an unusual adenovirus that we don't all have, we haven't all been infected with before. And so as a result, when they give you that vaccine with that virus, your own immune system lets the virus replicate for a little bit. And when the virus replicates, the virus manufactures the spike protein, which is the target of all these vaccines. And you have an immune response to the spike. If you were to use an adenovirus that we've all seen before, then the virus wouldn't replicate, and so you wouldn't make that spike, and you would never have a good immune response. So that's the J&J vaccine. Word on the street is the J&J vaccine is probably going to get approved during the month of February, so it probably will be available in the US before too long. The other adenovirus is this Oxford AstraZeneca adenovirus. You may have heard about that one too. The difference is that adenovirus was obtained from a chimpanzee. So no humans have immunity to it. So along the same reason, for the same reason I've just mentioned, it can replicate, and then you can react to the spike protein and generate immunity. At this point, the AstraZeneca Oxford vaccine takes 2 doses. The J&J vaccine may only require 1 dose, which would be a big advantage. The other advantage of the adenoviral vaccines is they're very, very easy to manufacture. They were a little bit harder to make because you've got to put the nucleic acid instructions into a virus rather than just making it like we did with the RNA or the DNA vaccines. But once it's in that virus, viruses grow in cells at a very, very fast rate. And so you can make potentially billions of doses of vaccine in a relatively short amount of time with adenoviruses. So they weren't the first ones there. RNA is the first one. They're probably going to be the third and fourth ones across the finish line. And then the traditional viruses, which the examples would be, say, Novavax. You may have heard of Novavax. They were one of the early companies that got lots of US government money. Novavax will probably be the 5th one. And then the Sanofi vaccine, which they're developing in collaboration with GSK, will probably be the 6th one. My company, we compete with Novavax and GSK. We'll probably be about the 30th one or something. We are not going to be at the front of the race. We're going to be the sort of next generation vaccines that will hopefully be better, but we weren't as fast and we did not get to the front of the queue. So that's a history story. Now let's get to what do we know about these vaccines. Lots of people are very concerned about the difference in time for developing these vaccines compared to the traditional route. And there are some logical reasons to be concerned about that. There are also some reasons that maybe we shouldn't be concerned about it. So let me just walk through those reasons. You remember when I said we approve vaccines based on efficacy. So both of these RNA vaccines, that is the Pfizer vaccine and the Moderna vaccine, have been shown to be efficacious. And just to give an example of that, I've got a slide here from the FDA's review of the Moderna vaccine. And the red dots are cases that occurred in people who received the Moderna vaccine, and the blue dots are in people, the number of cases in people who received placebo. So you can see there's a dramatic difference in the number of cases in people who got placebo versus people who got the vaccine. So both these vaccines clearly are effective, they're efficacious, they prevent people from getting disease. The second topic we talked about for getting approval was tolerability. And both of these vaccines are somewhat irritating. Let me just find my slide here for a second. And when I say somewhat irritating, many of you may have, who are my age or older, may have received the shingles vaccine from GSK. That vaccine is quite poorly tolerated. About 80% of people will develop a sore arm, fever, fatigue, and the like after they have received that vaccine. And of course, now I can't find the slide I wanted to show you. There are other vaccines that are very well tolerated where not very many people develop a sore arm or fever or fatigue on the days after vaccination. These are on the more reactogenic side. About 50% to 75% will have a substantially sore arm the day after they get the immunization. And the way we typically measure how sore your arm is by asking you what activities you're able to do. So an example would be, if you can't move your arm the next day, we would call that severe. If you are trying to, if you can move your arm, but you can't, say, comb your hair, we say, well, that's a moderate reaction. If you notice that your arm is sore while you're combing your hair, we say that's a mild reaction. If we talk about how sick you might feel the next day, if you can't go to work, we say that's a severe reaction. If you can go to work but you don't feel very well, we say, okay, well, that's a moderate reaction. And if you're just fine, but you notice, well, I don't feel quite as zippy as I normally do, we call that a mild reaction. For the Pfizer vaccine, about half the people report mild to moderate pain in their arm on the day subsequent to vaccination. And about 10 to 20% will report fever or not feeling well on the day after vaccination. And the Moderna vaccine is a little bit worse, but not substantially worse. So if you get a choice, again, I'm not giving medical advice, I'm giving my personal opinion. If you get a choice, the Pfizer vaccine appears to be better tolerated than the Moderna vaccine, and they're both equally effective. So while I would take either one, I would prefer to get the Pfizer vaccine rather than the Moderna vaccine. So when these two vaccines got approved, they both had been tested in a trial involving between 30 and 40,000 patients, which meant 15 to 20,000 patients got the vaccine. Remember when I said traditionally we treat 3,000 to 10,000 people? So actually more people have received these vaccines at the time they were approved than we would normally treat. Now there has been one previous vaccine in the early 2000s. Merck developed a vaccine for rotavirus, which is a cause of diarrhea in children. That vaccine had previously been taken off the market for safety concerns. So in that context, where there were specific things to be worried about, the FDA required them to treat 35,000 people before they would approve that vaccine. We're in a different situation here, and so they haven't asked for 35,000 people to be treated. But the point I want to make is we know a lot more about the immediate tolerability reactions for these vaccines than almost any other vaccine that's ever been developed. The thing we don't know about these vaccines is the long-term follow-up. For both of these vaccines, the longest people who'd been followed at the time the vaccines were approved had been followed for a total of 6 months. So instead of having 3,000 to 10,000 people followed for at least 12 months, here we've got the maximum people followed for 6 months, and some were followed for as little as 1 month. So the average was about 4 months. So we don't know as much about long-term safety of these vaccines as we know when most vaccines get approved. To some people, that's quite disconcerting, and people have to make their own decisions about that. My perspective is we know that these vaccines are safer than getting infected, and therefore I would take the vaccine, but we need to be clear with people that the long-term safety of these vaccines has not been as well studied as it is with most vaccines when they get approved. We know more about the tolerability, but we know less about the long-term safety than we routinely do. That's kind of the end of what I was planning to share from a general overview for COVID vaccines. I'm happy to answer questions if you want to put them in the chat box. We had some that were submitted prior to the meeting, so I've actually got a list of some here as well. But the main thing I want to try to communicate to people is that we know these vaccines work, and we know more about the short-term safety issues of these vaccines than we have for almost any vaccine we've ever developed. What we don't know about is the long-term safety, and the long-term safety, to me, I consider it to be much better than the long-term safety of being infected. So with that, I'm happy to answer any questions that you might have.
Yeah, so we will go ahead and open it up for questions. Like Dr. Martin said, if you do have a question, please go ahead and put it into the chat. We have a few that we're going to address here that were submitted prior to today. So we'll address a few of those, and then we'll take your other questions. So the first question, and you just answered this, is should I be concerned with the long-term side effects with this vaccine being rushed to the market? Yeah.
So again, just to repeat what I said, we absolutely should be concerned, and the FDA is continuing to require the manufacturers to follow these patients to assess safety over time. But I think it's clear at this point that we're months after these patients have been immunized, and we're not seeing problems, and that it's better to get immunized than it is to get infected.
The next question is it safe to get the vaccine if you have already contracted COVID-19 last year and have the antibodies in your system?
So at present, we believe it is safe, but there are some important caveats to give to that answer. So in the study where we looked at these 40,000 or 50,000 people in the two different studies, those were people who did not have COVID prior to their coming into the study. So we don't know how the safety of these vaccines really is in people who have COVID infection. Now, as we've rolled them out, and we've now treated about 10 million people in the United States, many of those people will have been previously infected because we don't test people now before you get the vaccine to say, have you been infected or not? We just immunize people. So it is the recommendation is that if you have been infected, you should still get the vaccine. In some states, because this immunization policy is being handled at the state level, some states say if you have been infected and you're known to have been infected, as you actually had a positive test, not just that you had symptoms that you thought were COVID, but you had a positive test, they have you go to the end of the vaccination line because you will have some immunity from the infection. So you're not as susceptible as an unimmunized person who hasn't been infected. So different states are doing it in different ways. At this point in time, my understanding is that the state of Nebraska is not going to make people who were previously infected go to the end of the line, but in some states they are.
Okay. If someone has had an adverse reaction to flu shots in the past, should they be concerned? For them, should there be a concern about getting this vaccine?
So the major reaction that's of concern for this vaccine is a reaction called anaphylaxis, which is a very severe allergic reaction. Basically, people stop breathing. At this point, it seems to be happening about 10 times more common than it does with most other vaccines. And we're not quite sure why that's the case with this vaccine. The recommendation is that if you have severe allergic disease, you should not take this vaccine. You should wait. So depending on what kind of reaction one's had to a previous flu vaccine, it would depend. If your reactions to the flu vaccine were things like your arm got sore or you felt sick the next day, you should still take it. Your arm will probably feel sore and you will probably feel sick the next day, but you would still benefit from the protection that the vaccine would confer.
This next question is, I'm currently breastfeeding. My son is 4 months old. I've read that there were few, if any, breastfeeding women in the vaccination trials. What factors should I consider when deciding whether to get vaccinated while still breastfeeding?
Yeah, that's an excellent question. And I am going to slightly divert from the standard recommendation here in what I'm about to say. So I want to be clear, I'm giving my opinion. This is not medical advice for anyone. We've tended to lump, when you look at the CDC policy, they've lumped pregnancy and breastfeeding into a single bucket. And I think that that's actually not appropriate. So I would be reluctant to take this vaccine if I was pregnant. I would not be reluctant to take the vaccine if I was breastfeeding. If you're breastfeeding for the RNA vaccines, you might express antibodies in your breast milk. I think that's a good thing, not a bad thing. But if I was pregnant, lots of vaccines, we have only limited data about use in pregnancy. There are some we have quite a bit of data, but most of them we don't have much data. So I would probably be waiting until one of the traditional vaccines came along if I was pregnant. So I want to make a distinction between pregnancy and breastfeeding, which is not actually what you'll see on the CDC guidance. The CDC guidance puts them together, but I think they're actually two very different situations.
This is a follow-up question. Do vaccine candidates normally test pregnant women at all, or do they simply go off the basis that it was safe in a whatever mammal, then it will also be safe in the humans as they were also mammals?
Yeah, that's an excellent question. So it's a rather complicated answer. So all vaccines and all drugs have to have testing done in animals that we call reproductive toxicity. So you test your drug in pregnant animals. You look at the progeny from the pregnant animals. You'll often test it in the progeny, and then have the progeny get pregnant and look at the next generation. So actually go for 3 generations to look for toxicity from the drug or the vaccine that you're trying to test. And we don't if you get a positive result in that test, you've really got a problem with your drug or with your vaccine. Because, finding a problem is, say, if you treat 10 animals and you see a problem, that means you've got a 10% problem rate. That's a big problem. So it's not super satisfying to know that there aren't problems based on a reprotox study. When we actually do the clinical trials, because we don't know anything about the safety of these vaccines for pregnant, when we actually exclude pregnant women from the clinical trials, and in fact, we require that people use contraception during their participation in the clinical trials. So we want to prevent pregnancies from happening. Now, as you can imagine, advising people to use contraception and actually having them effectively use contraception are 2 different things. So there will inevitably, in any development program, be people who become pregnant while they were taking the experimental drug or vaccine. When that happens, you follow that person to see exactly what happened and if there's a problem or if there's not a problem. Usually there's not a problem. But the point is, when your drug gets approved, you really don't know very much about its use in pregnant women. Every drug or vaccine then undergoes what's called a registry, that if it gets used and somebody subsequently becomes pregnant, or perhaps they got treated while they were pregnant but they didn't yet know it, all those people go onto a registry to collect data to say, okay, well, here's somebody who accidentally got exposed to this drug or this vaccine while they were pregnant. What happened? And all those pieces of information are collected. And at the end, someone will eventually be able to say, okay, we've now inadvertently used this drug in 1,000 people and there aren't any problems. So now let's actually say that it can be used in pregnant women. But it's actually a very long time before most vaccines or drugs are labeled safe for pregnant women. Now, the one exception to that is if you're developing a vaccine for use in pregnant women, well, then obviously it's going to be a whole different story. You're going to, from the very beginning, be testing it in pregnant women, but that's not what we typically do with most vaccines. So that's a really long answer to that question, but it's actually not a super straightforward answer.
Okay. So far, the vaccines have been distributed. The vaccines being distributed are only for adults ages 16 or 18 and older. Why were no vaccines created for children, and are there vaccines being developed for those under 16?
Those are great questions. So the first reason why they weren't initially developed in children, we have a rule when we do clinical research that you have to obtain informed consent. And so we consider it unethical to test a drug in children that hasn't already been tested in adults, because as an adult, I can give informed consent to test your drug on me. But if I'm a child, it's the parent that gives informed consent. So until you've demonstrated that your drug is reasonably safe and effective in adults, we think it's not ethical to give it to children. Now, in this situation, again, we're in an emergency, so people went pretty fast. There are trials ongoing right now for children, and I would imagine that one or more of these vaccines will eventually be approved for use in children, probably sometime during the summer. But the focus was initially on adults, and in particular older adults, for two reasons. One is this ethical reason, and the second one is that's where the deaths were happening. So the priority to get an effective vaccine out was to prevent death, which is happening in older people.
After I get both COVID vaccines, because there are 2 doses, what precautions do I need to take?
That's a great question. We don't know the answer. These studies that I mentioned that both Pfizer and Moderna did were looking at the frequency of disease. What we don't know is in those people that got disease, if you got the vaccine, did you have less viral shedding than people who got the placebo? Or were there other people who were infected and maybe shed a little bit of virus but didn't get sick? We don't know the answers to those questions. The studies weren't designed to answer that question. So the current recommendation is that after you get vaccination, you should still, or after you receive your vaccination, you should still social distance and still wear a mask like we've been doing up until this point in time. When most of the population has been immunized or infected, then we can sort of relax a little bit. But at present, because I don't know if I still shed having received the vaccine or not, I put my neighbors or contacts or family members at risk if I don't follow the same precautions that we're following now.
What is the current understanding of vaccine effectiveness and side effects for people with autoimmune diseases?
That's another great question. And I'll say that it's limited. So let me tell you the general concern we have about vaccines and autoimmunity. Autoimmunity is where something has gone wrong with our own immune systems and they're now reacting against ourselves. Normally, our immune systems tolerate ourselves and we react against foreign things. That is, things that our body sees that aren't us, and we don't react to us. People with autoimmune disease, something's gone wrong with their immune system, so they now react to themselves. And the concern when you give a vaccine to a person with autoimmune disease is that with that stimulation of their immune system, might you also cause an exacerbation of their autoimmune disease. People with autoimmune disease were not enrolled in either of these trials. So we don't have any data about exacerbations. People who are now being immunized are not being excluded based on autoimmune disease. In fact, there are some reasons to think people with autoimmune disease might be at more risk of complications from COVID infection, because people with autoimmune disease often take immunosuppressive drugs to calm down their immune system so it doesn't react against themselves, which might put them at higher risk for complications if they get infected with COVID. So people with autoimmune disease, it's actually both a more dangerous situation to get infected, and we don't know as much about the safety of the vaccines in that particular group. So it's actually a more open question for people with immune disease than it is for other adults. Now, all that said, my wife has an autoimmune disease and she is on an immune suppressive medication. It's our intention to get her immunized as soon as she comes up on the list, because I still think the risk of infection is worse than the risk of vaccination. But it's a complicated question.
Let's see, these are a few questions from the chat. Could you comment on adverse effects people have had from the vaccine?
Well, so Carol, thank you for that question. The main adverse event that is really scary for people is this anaphylactic reaction. And because of the increased frequency of this reaction, that's why they want you to stay in the clinic for 15 minutes after you've been immunized, which is slowing down the queue for immunization, because you don't just pop them and go. You have to get immunized and sit there for 15 minutes. So the queue takes a while to process. The treatment for anaphylaxis is an injection of epinephrine. So you may have heard about these EpiPens that were, in the news a few years ago for people with peanut allergies. That's the same treatment that when kids with peanut allergies have a bad reaction, it's the same reaction. It's this anaphylactic reaction. So the treatment is epinephrine, and it's fairly quickly reversed if you get epinephrine. If you don't, though, it can be fatal. And so it's the reason why these vaccines are only being given in these sort of, fairly highly controlled environments because of this risk. It's a low risk, but it's routinely about 1 in a million. And in this case, it's been more like 10 in a million. So that's high enough, that you don't want to take a risk and let somebody, say, have a drive-through clinic where somebody gets immunized and then 3 minutes later they have an anaphylactic reaction while they're driving down the highway, and it could be a bad situation.
You had mentioned earlier that we know that there's 2 out there being distributed right now, and there's several others in the works. Would there be a benefit to waiting for the vaccines that are almost ready to go?
Yeah, that's an interesting question. I would say, I'll just again give my own opinion. My own opinion is I want to get the Pfizer and Moderna vaccine as soon as I can. I'm a physician, but I do research. I don't take care of patients, so I'm not frontline risk. I'm not in the ICU taking care of sick people. I'm sitting in my laboratory doing experiments with mice. And by the way, mice don't get infected with COVID or with SARS-CoV-2. So I would take the approach, unless there was some particular reason or some peculiar reason that would be better to wait. I wouldn't wait. I would get it as soon as it's available in my area. Now, I know in some parts of Nebraska, and again, I can't speak to other states because I don't know what's happening on the ground in other states, but in some parts of Nebraska, all of the healthcare workers have been immunized, and they are now immunizing older people or people with underlying medical conditions. In Lincoln, that's not the case. We haven't finished immunizing all of our healthcare workers. We've probably got 2 or 3 more weeks to go before we're done with that here in Lincoln. But if I lived, so for instance, my aunt who lives in McCook has already received her vaccine. So the McCook area was already immunizing older adults, at least last week, because I got a question from her last week on it.
How will new strains of the virus affect the vaccine efficacy?
That's another great question. So to date, so far, there was just data that came out today on the Pfizer-BioNTech vaccine from a study done in the UK. These variants, while they can be measured genetically, because they have a different gene sequence, they don't have a different immunologic profile. So our antibodies generated to the vaccine still react to the variant viruses. We don't know how long it will take before a mutation develops that won't react with these with the antibodies that we've already generated. That's what happens with flu, and that's why we have to change the flu vaccine all the time. At this point in time, it looks like the SARS-CoV-2 is a more stable vaccine than influenza. So we're hopeful that this might kind of be a one and done kind of a thing, but we just have to keep our eyes on it. We haven't seen it long enough to really know how it's going to vary over time.
what can be some of the long-term safety concerns?
Yeah, well, so let me just mention for regular vaccines, for all the other vaccines that we don't really have long-term safety concerns. We've looked for a long time for long-term safety concerns, and we don't see them. It was part of the reason why the FDA said that they wanted to see 2 months of observation, because we do see things that happen up to 2 months after you get a vaccination. The kind of the end of the window is 6 weeks, but we don't have any vaccine that's had a safety signal happen from 6 weeks to that 12-month period. So while that's our standard follow-up, and we like to have it so that we can say to people, look, we looked for 12 months and nothing happened. The real window is within the first 6 weeks after vaccination. Of course, the real window is in the first 7 days. So that's the kind of thing that we're interested in. And of those things that happen in the first 6 weeks, the one that happens the longest is a reaction called Guillain-Barré syndrome, which is a immune reaction against your own nervous system. So some of you may remember in the 70s when we had a swine flu epidemic, and there was a swine flu vaccine was given, and shortly after vaccination, about 50 people in the United States developed a lower body paralysis following vaccination. That's what Guillain-Barré is. It can happen after any viral infection, but with some vaccines, it's known to occur within, say, 6 weeks of infection. So far, there have been no cases of Guillain-Barré seen with any of these vaccines, but we have to keep looking because it's a rare event, and sometimes you don't see rare events when you've only treated a few thousand people, a few tens of thousands of people.
And we will take a couple more questions. If you have a question, make sure you add it to the chat. You had mentioned that in the past, mRNA vaccines did not work. What is the difference this time, and why is the mRNA effective for COVID?
Yeah, Gretchen, that's a really good question. It's one that people like me that work in this vaccine research space are really interested in. So there's going to be one of two answers to the question. Either it's going to turn out that SARS is a really easy virus to stop from an immunological standpoint. So while it didn't work for other viruses that they've looked at before, like flu, maybe SARS is just really much more easier for our immune systems to stop than flu, and therefore these vaccines could generate enough antibody that worked. Maybe they'll never work for anything else, but they worked here, and thank God for that. The other possibility is maybe these vaccines are going to be the vaccines of the future, and maybe they just were tested in the wrong way or not the right dose or something else for why they failed before. So lots of people are kind of trying to figure out, is this really the wave of the future, or is this, we were lucky that SARS-CoV-2 could be inhibited so easily. We'll find out as the other vaccines come along. 95% vaccine efficacy is really quite incredible. So if it turns out that all these other vaccines get 95%, then that would be one thing that might say there's nothing so special about the RNA. But let's say that the other vaccines all end up at like 60% or 70%. Well, then we might say there actually is something very special about these RNA vaccines. And maybe we should move in the future to all vaccines being RNA vaccines, or at least many vaccines being RNA-based vaccines. This is a real controversy in the vaccine world today, the vaccine world that I live in.
Another question here: if you decide to wait to get the vaccine because you are currently because of a current pregnancy, will there be an opportunity for that person to get it later this year after giving birth?
Well, I would say yes. I don't think we're going to be done vaccinating people until, say, the end of the summer. So depending on on the timing of the start and the end of your pregnancy, that may be a little bit more complicated. I just can't imagine that all of a sudden there won't be any vaccine available anymore. I'm sure this is going to go on for a while, and we'll still have some laying around. The real question is, how long are these vaccines going to last? And for that, we'll just have to wait and see. Do they last for 1 year, for 5 years, for 10 years? It would be nice if they turned out to be like some vaccines we give, where they really last for a lifetime, or at least they would last, say, for 10 years, like a tetanus shot does, as an example. Hopefully they won't be like flu, where you have to get one every year. But the truth is, this disease has been around for a year. We have no idea how long either the natural immunity or the vaccine-induced immunity is going to last.
Okay. Is there any other questions out there for Dr. Martin? If so, you can add it to the chat or you can unmute yourself to ask. It's that weird, awkward silence. Oh, we got one in here. All right. I have seen published row values from 2 to 5 for determining herd immunity for SARS-2. Have you seen more consistent value in the literature?
So, so Trish, that's called R naught in this case, and it's sort of a measure of how many people get infected from an index case. And I'll say that there's a little bit of a misconception about that number. It's not a fixed number for each virus. It's a fixed number based on the virus and the epidemiologic setting for that virus, i.e., as an example, if there are lots of immune people around you or not so many immune people around you. So there isn't really a specific number. Social distancing reduces the R naught. Wearing a mask reduces the R naught. So it's not really a number that we can say, oh, this is it. It's a number that depends.
Okay. And we did talk about this earlier, but when are students likely to receive the vaccine?
So I will make a distinction between two different kinds of students, college students and younger than college students. College students, my guess are they'll probably be starting to get immunized, say at the end of this semester and over the summer. Younger students than that, my guess is they'll start to get immunized maybe late in the summer. And they'll probably be getting immunized through the school year. The difference for students as compared to teachers is students tend to not have the same complications from infections that teachers do. So that's why in most states, teachers are in the next wave that's going to be immunized after the healthcare workers, because they are essential personnel and they're exposed. They're in situations where the exposure rates can be high. Kids have the same exposure, but they don't have the complications. So kids will probably go to the end of the queue.
Okay, and last question, does double masking have a valuable effect?
Yeah, so I guess it depends on your definition of valuable. I personally don't double mask, but I do anytime I'm not either sitting at my desk like I am now or at home with my wife. I wear a mask outside all the time, including if I go to the coffee machine, which is just out around down the hall from my office. So I do think that wearing a mask is important. If one thinks about it, 90% reduction from wearing a mask of droplet transmission, wearing 2 masks might be another 90% drop, but that's a drop from now at 10% risk to 1% risk. It's not a substantial improvement. Now, there are some people where reducing the risk from 10% to 1% might be important for them. So I'm not saying people shouldn't do it, but I am saying I don't recommend or demand, or in our office, we don't require anything like that. We do require that we all wear masks if we're out in the common areas of our office building.
Okay. I had one more come in. Someone that tested positive and was fortunate to get IV BAM.
Yeah.
I think it helped me with my recovery. Heard I need to wait 90 days to get vaccine.
Yeah, that's a good question. I frankly don't know the answer to that. So this is a person who got one of the monoclonal antibody treatments for COVID. So the kind of thing that some of the politicians have received when they've gotten infected and it's made the news. They're good treatments, but they do now give you COVID antibodies because they are antibodies against COVID. So there's a concern that they might interfere with the uptake of the vaccine. That is that if the antibody binds the target before you can generate your own antibodies to bind the target, you may not have a good response to the vaccination. So I don't really know what the answer is to the question. My apologies, but it's one that would probably be a good idea to ask your physician or your pulmonologist or ID physician that gave you the antibody what their advice would be.
I think that wraps up our questions. Thank you so much to all of you for joining us tonight. And thank you, Dr. Tyler Martin, for your time and your expertise. We truly, truly appreciate it.
Thanks, everybody. I had fun. I hope it was helpful.
Absolutely. Lucas, I will let you wrap it up.
I just want to echo thanks, Tyler, for your time and your really important work. I mean, I think it's great for us as alumni to hear from you, someone who's worked with this, and I hope everybody finds it valuable and useful to their personal decision making. You know, we're glad to do these things as an alumni association. The way we associate with our alumni's changed this last year. It's you know, I think we've discovered new ways of connecting, and there's no other events where we can have 16, 17 states in the same room per se. So this is great. Thank you for joining us. You're very important to us as alumni. What you say about the institution is helpful to us. You know, there's different ways for you to get involved. You know, one of the things that we're working on right now is something called the Dream Team. If you have an interest in helping us recruit students, that's a very important service to the university. Without those students coming in, there's really no reason for us to do any of this other than it's just fun. Your advocacy with legislators in our state is important as they make decisions about funding for the university, and your financial support's important too. We treasure you. You're among the 55,000 living alumni around our country, around our world, and we're so appreciative of what you do for the institution. So with that, I look forward to seeing you sometime soon, hopefully face to face. Again, thanks again to Tyler. And with that, we'll bid you a good evening. Thank you.